So now I think it's time to move on, and I'll turn it over to moderators Dr. Katie O'Brien and Dr. Shine Chang.
Hi, everyone. Welcome. We are so pleased to have you here for the first session, which is going to focus on COVID-19 research in cohort studies. It's an exciting topic, and there's news coming out every day, every minute almost, and we are looking to the future because we know the future is going to be better, much, much better. So we have planned a 30-minute presentation to be followed by a moderated 20-minute Q&A session. Please use the chat function to submit your questions. Please also keep yourself on mute in case of interruptions. The session is being recorded for your viewing pleasure later on. And with that, I'm going to turn it over to my session co-host, the very excellent Dr. Katie O'Brien from NIEHS, who will introduce herself and our speaker. Katie.
All right, welcome everyone. Nice to have you here today. So I'm Katie O'Brien. I'm a staff scientist in the epidemiology branch at NIEHS, and I'm part of the cohort consortium through the SISTER study. And it's my pleasure to introduce you today to Dr. Jeffrey Ginsberg, who is the director of the Center of Applied Genomics and Precision Medicine at Duke University School of Medicine. He also has several other appointments within Duke, including the School of Engineering, the Department of Medicine and Pathology, and the School of Nursing. So Dr. Ginsberg is the founding director of the Center for Applied Genomics and Precision Medicine at the Duke University Medical Center, and for MedX, a partnership between the schools of medicine and engineering to spark and translate innovation. His research addresses the challenges for translating genomic and digital health information into medical practice and the integration of precision medicine into healthcare. In 2017, he received Duke's Translational Research Mentorship Award and is a finalist in the NIH BARDA Antimicrobial Resistance Prize. He was a recent member of the Advisory Council to the Director of NIH and is co-chair of the National Academy's Roundtable on Genomic and Precision Health, and is founder and president of the Global Genomic Medicine Collaborative, a not-for-profit organization aimed at creating international partnerships to advance the implementation of precision medicine. He's recently served as a member of the Board of External Experts for the NHLBI, the Advisory Council for the National Center for Accelerating Translational Science, chair for the review of Genome Canada's large-scale applied research competition in genomics and precision medicine, and the World Economic Forum's Global Agenda Council on the Future of the Health Sector. He is the founder of Predigen Incorporated and MeTree and You Incorporated. He was previously vice president of the Molecular Medicine at Millennium Pharmaceuticals Incorporated and a faculty member at Harvard Medical School. Dr. Ginsberg is the founder and co-chair of the Global Genomic Medicine Collaborative, a not-for-profit dedicated to implementing genomic medicine with a focus on low-resource setting around the globe. The G2MC operates the International 100K Plus Cohorts Consortium, of which Dr. Ginsberg is a co-chair, along with Terry Manolio and Peter Goodhand. And with that, I'll turn it over to Dr. Ginsberg to tell us about his work with the 100K Cohorts Consortium.
Thank you very much, Dr. Shang and Dr. O'Brien, for that introduction. I'm very grateful to be here for the opportunity to talk to you about the 100K plus International 100K plus Cohorts Consortium, or IHCC. At this NCI Cohorts Consortium, it's really nice to have a chance to see Cohorts Consortia to Cohorts Consortium bidirectional dialogue taking place. As Dr. O'Brien said, I'm Jeff Ginsberg, and I'm here on behalf of my co-chairs, Terry Manolio of the National Human Genome Research Institute, and Peter Goodhand, who is the executive director for the Global Alliance for Genomics and Health. May I have the next slide, please? The concept for this consortium arose from the notion that for decades, large cohort studies, as the NCI consortium exemplifies, have been established worldwide, and that a significant amount of international investment has been made in these cohorts to amass huge data sets. But because of geographical boundaries, constraints in ancestral origins of these cohorts, and even size, the types of opportunities for analysis are somewhat limited. And therefore, we reasoned that combining data from these cohorts would allow them to address pressing global health questions that no one of them can answer alone. This would enhance the value of each one of them, and obviously leverage the enormous investments that the funders have made in them, and it would create a force amplifier for scientific investigation. Next slide, please. The vision for the IHCC is to create a global network of large cohorts with multidimensional data from diverse populations that will be maximally utilized to enhance scientific understanding of the biological, environmental, and genetic basis of disease in order to improve population health. This vision was really meant to focus both all on discovery, on translation, and ultimately on population health sciences. Next slide, please. The brief history of the IHCC is here. In 2015, the National Institutes of Health began compiling information on large cohort programs that were going to assemble greater than 100,000 participants. And in part, this was with an eye towards really understanding how to best leverage the All of Us research program, which, as you know, aims to have 1 million participants. In 2017, the heads of international research organizations, of which Francis Collins and Jeremy Farrar from the Wellcome Trust were co-chairs, agreed to bring the cohorts together to encourage data sharing and to maximize many of the HEROES investments. So the first summit was held in March of 2018 at Duke University. The second cohort summit was in April of 2019 in Iceland. And then this past May, if you've heard, we had a third virtual summit, of which the NCI Cohorts Consortium was an attendee. Both the G2MC, the Global Genomic Medicine Collaborative, and the GA4GH, the Global Alliance for Genomics and Health, were asked by the HEROES to really make this happen, given the global nature of both of these organizations and their focus on not only genomics, but on data standards. Next slide, please. So as I mentioned, the first International Cohort Summit was held at Duke in 2018, and this was clearly an exploratory meeting to understand whether there was even enthusiasm for the cohort leaders to engage. So we really discussed at length the prospects for collaborative opportunity and the compatibility of cohorts. We discussed also at length the concept of a registry of cohorts, which I'll come back to a little later. And we began to look at and understanding some of the initial barriers to data sharing and specimen sharing. And from the outset, we set 4 criteria for participation, particularly in this first meeting. One is that cohorts needed to have 100,000, aspire to have 100,000 or more participants. It was unselected for disease, have available biospecimens or the capability to collect them, and the potential for longitudinal follow-up. But in selected cases, we agreed that we would consider cohorts that had a unique demographic, that would have unique ancestral origins, and particularly cohorts from low and middle income countries, or low resource settings, which were largely unrepresented particularly in genomic databases, but potentially in other databases, that we would relax these criteria. Next slide, please. So in March of 2018, we had 100 attendees from 24 nations representing 60 cohorts and 30 million participants. And the attributes for these can be found on our website, which I'll come back to at the end. It's ihccglobal.org. Next slide, please. But we certainly recognized from this first gathering that there would be significant challenges to combining cohorts in a meaningful way. First of all, there's missing data, and the complexity of limited documentation would exist. There would be also complexity in documentation of available data. We recognize that there was also extreme heterogeneity of the way the data were collected, or the variables that were collected, and this results in really a lack of standardization or maybe a challenge to harmonize questionnaires. There were known to be international laws or regulations that might prohibit moving particularly samples across international borders, but potentially data as well. There were certainly, admittedly, lack of standards for phenotypic information and health outcomes that were documented either in the cohort data or in associated electronic medical records with which they were associated. And at the participant level, there was potentially limitations on data sharing and access, given the potential for cultural differences in risk tolerance and demands for privacy. Next slide, please. But we persevered or pressed on and thought there would be potential solutions to many of these challenges, that particularly with the Global Alliance for Genomics and Health, that we would be able to consider solutions that would articulate data standards, that we could standardize or at least consider the consortium to adopt standards for data collection using tools such as the Patient Reported Outcomes Measurement Information System or the NIH's Phoenix phenotypic database, which we think would add significant value to the standardizing phenotype. We were very cognizant of the data, of the ability to create a centralized database, and we really thought that the primary strategy for this consortium would to use federated data analysis. So we would move the analysis to the data rather than vice versa, but recognizing that might not always be the case, in all likelihood, we would have a hybrid solution. We would encourage standardization of computable phenotypes, particularly coming from electronic medical records. And in cohorts which did not have access to digital health devices or wearable devices or certain apps, that we would enable and facilitate the uptake of some of those phenotypic technologies into cohorts that lack them. And where possible, we would tap into already existing frameworks such as the GA4GH or All of Us research programs frameworks for privacy, security, and consent. Next slide, please. We also began to dream about the science that could be accomplished as we undertook this very large and potentially productive collective consortium. Here are some of the ideas that were brought forward at the first summit to be able to look at extremely rare exposures and outcomes and the risks associated with them, given the scale of the data sets we're talking about, to begin to really understand the generalizability of risk factors developed in one population in others, to develop more population-specific determinants of health, social or cultural determinants of health, and even country or cohort-specific risk models. And I think one of the most ambitious ideas that came out of this first meeting was to create a catalog, a complete catalog of all human knockouts with associated phenotypic information, something that could only be done at significantly large scales of the type we were talking about. Next slide, please. So in 2018, we moved forward with 3 goals and 3 work streams. One was to develop the information technology that would allow for discoverability of the cohorts and sharing of data. So this would be essentially a way to de-silo the cohorts. The second would be to come up with a scientific plan that really got the scientific community excited and that leveraged cohort collaborative opportunities, as well as interchange with scientists from around the world, and where necessary to take a leadership role in a policy agenda that would allow for data sharing, as well as engagement with industry. Next slide, please. So last year, even though it seems like a much longer time than just a year ago or so, we were gathered in Iceland, home of the primordial cohort. And we had 117 representatives of 67 cohorts, 29 countries. We also had significant involvement from the heads of international research organizations whose acronyms are listed here. And we agreed that we would begin to move forward with finalizing our information technology and policy agenda to support the science, that we would engage in at least one pilot demonstration project to show that this could actually be done, and to develop a more formal charter that would define the basis for our ways of collaborating, including rules of engagement for biopharma and technology firms. Next slide, please. So as I mentioned, one of the areas of import was to do a proof of concept study that would actually illuminate the ability for cohorts to actually do collaborative science at scale. So what came out of the Reykjavik meeting was the notion of creating a polygenic risk score. This was led by Children's Hospital of Pennsylvania, Patrick Sleeman and Hakan Arasan. And the types of phenotypes for the PRSs are listed here. They were not, these are common attributes that would be hopefully available in many of the cohort data sets: asthma, blood pressure, BMI, type 2 diabetes, and height. The CHOP group created unique weight files for the causal variants and sent that out to the cohorts for analysis. 8 cohorts participated from 5 countries. The transethnic scoring outperformed all population-specific scores for the non-European cohorts with similar predictive values. And really, this was meant to be a feasibility study, which we deemed as successful in showing that the federated approach of generating scores at each site could be done. While there were challenges, I think they were we would be sufficiently able to overcome them to do future analyses and a much more compelling scientific questions. The next slide, please. So a very exciting part of our work has been the development of the IHCC Cohorts Atlas. This is really meant to bring tiers of data together in a very nice, hopefully a nice user interface, starting with cohort name and web links to metadata, eventually to go deeper into data methods and data types, and probably a long-term aspiration might be to have individual type of data. The Atlas is really aiming currently to begin to collate the data dictionaries of all IHCC cohorts and harmonize them, and to create an online user interface. It's been spearheaded by the European Bioinformatics Institute and the Ontario Institute for Cancer Research. That's Thomas Keane at EBI and Philip Awadallah at OICR, and their teams have really been instrumental in putting this together. Next slide, please. So this is just a short schematic of the process of bringing together the cohort data dictionaries, administrative data into the OWL semantic files on the left-hand side, and really using efforts that were previously ongoing from Synica, which is a consortium of biobanks in Europe, Canada, and Africa, whose goals are to develop tool analysis, which is a data analysis consortium, and really do the mapping step, and eventually create the atlas once all these ontologies are mapped. So on the next slide is I wish I was technically proficient enough to actually show you a live demo, but I have a couple of screenshots just to show you the atlas. So this is the splash page you'll see when you go to our atlas. On the left-hand gutter is a series of attributes of the cohorts. You can see the name. There's a tab for the name, countries, biospecimens, sample types, current enrollment, the names of the PIs, behavioral measures, physiologic measures, and demographic and economic outcomes. In the middle is sort of the display of what you would see once you sort of when you put in your search criteria. And you can see also there's a nice place for reference so you can find out who's really in charge of these cohorts and directly contact them. I have a screenshot up next, I think, of a search on South African cohorts. This would be the two in the database at the time, South African Research Population Infrastructure Network, or SAPRIN, and the African Health Research Institute's cohorts. You can see the way the display is, shows you high-level attributes of those cohorts. And in the upper right-hand side is a schematic of the kinds of biospecimen data that one might find. If you can toggle to the next slide. So this tells you that urine specimens are available in the African Health Research Institute cohort. Next slide, next, yeah. And then you can see that stool is available in both cohorts, and next, saliva is available only in SAPRIN. And that's kind of the graphical user interface that we're developing for biospecimen displays, and there'll be more progress on that hopefully in the coming months. Next slide, please. Also, as we were leaving Reykjavik, we thought it was really important to have policy deliverables on data sharing, on governance, on consent. We've achieved most of those. General data protection regulations is something we're still sorting through. And also we wanted to have a better articulation of the value proposition for participating consortia, as well as a policy for how we would engage with industry, biopharma, and technology firms. Next slide, please. So just briefly, we've adopted the GA4GH ethical framework, really focused on the individual participant and how they might benefit from research. Again, I'm not going to go through this in any detail whatsoever, just to say that we're we're committed to not having to reinvent the wheel. We really want to take advantage of things that have been done exceptionally well already. Next slide, please. We were to have our third international summit in Santiago, Chile, which is the sketch that you see in the upper right-hand side, but unfortunately, we were limited to a virtual meeting such as this one. But our aim was to really seek engagement of cohorts from lower resource settings, particularly in South America, Africa, and other regions of the world. We had 177 attendees, 27 countries, and 87 cohorts. We announced the charter that we had set out to do in a year earlier. We introduced the first working version of the cohorts atlas and our charter and governance documents. And we also thought it was really important to identify new and scientifically meritorious cross-cohort research projects. And of course, COVID-19 loomed large at that time as it does now. So we were implored, as you'll hear in a moment, by some of our funders to really take this seriously, particularly the Wellcome Trust from Jeremy Farrar and from Francis Collins. The next slide, please. So Francis was our keynote opening speaker at this meeting. Jeremy Farrar was the closing. I think, as you'll hear in a moment, they had really strong messages and words of inspiration for us of how to move forward, particularly in the context of COVID-19. So on the next slide is an excerpt from Francis's opening remarks.
How can IHCC help? Well, in some very important ways. When you look at the consequences of what happens to somebody who is exposed to this SARS-CoV-2, it's clear there's a lot of variability between the consequences. Some people who get infected but never even know it, completely asymptomatic but capable of passing the virus on to others. People who get very mild symptoms, and then people who get extremely ill. People who end up in ICUs on ventilators, and sadly, many of them also lose their lives to this disease. And we know that some of the risk factors are chronic illnesses and older ages, but that's not sufficient to explain the high degree of variability. IHCC is in a great position to be able to contribute to that because of your reach across the world. And certainly the focus that has been baked into this, which is to look at both environmental and genetic factors, means that IHCC has an opportunity to really identify with the appropriate power and numbers, what are the factors that play out in terms of risks of severe disease. And alternatively, what are the factors that pay out in terms of resilience? Why do some older people with chronic disease do just fine? What is that protection about? If we knew that right now, I guarantee you it would have all kinds of consequences for the way in which we're trying to prioritize drug therapies, because it's been proven over and over again, if you have a natural variation that provides some indication about a disease risk, that's a very good place to go if you're trying to develop a drug therapy. I wish we had that answer right now, but I know many people are starting to work on it. And more than that, I would say not just about susceptibility, but what about variations that play out in terms of drug responsiveness? I mentioned remdesivir and the fact that it does provide some benefit, but not equally to everybody. So what's that about? Is there some pharmacogenomics there that we ought to be looking at in terms of who responds well and who doesn't? And even with vaccines, which are coming along, as I said, in sometime in the next 9 months or so, are we going to be able to identify those individuals for whom the vaccine is particularly effective or particularly ineffective? Because I would be surprised, no matter how optimistic you are, if this vaccine is 100% effective for everybody. And we need to understand what those modifiers are in order to be able to adjust accordingly. So all of this seems to be like a great place for the IHCC to be positioned. And I know you're going to be talking about that in the course of this meeting with special session on COVID-19, indeed. We're even contemplating right now whether some of these cohorts might also be in a good position to be volunteers for testing out the vaccine, since they're often people who have been, at least in some of the cohorts, consented for recontact. And if we want to find out whether a vaccine works in various parts of the world, maybe this is not a bad place to think about starting.
Next slide, please.
How can I?
Francis Collins' comments now 6 months later still seem to be incredibly timely, both to our cohort and probably to yours. So it's really been terrific to have his support as well as that of Jeremy Farrar's from the Wellcome Trust. both really helping us move from our infancy to maybe early childhood or adulthood as a consortium. So following the virtual summit, we solicited applications for research funding. We were able to fund 6 proposals. The 3 that are listed in red on this slide highlight opportunities for COVID-19 research, and the other 3 are non-COVID related. I'm going to talk a little bit about the mental health and the South African COVID program in a moment, but as you can see, we also have a metabolic profiling, I would call that a cohort enhancement project to really explore the opportunity to look at metabolic profiling or metabolomics in cohorts. There's another opioid consortium that is seeking to organize opioid use data around the world to evaluate its long-term impact on cancer and cause-specific mortality. There's also an effort to look at obesity in infancy, childhood, and adulthood and assess its impact on cancer risk. So very germane to, I think, the NCI consortium, cohorts consortium meeting that we're having right now. And as you can see, there's a greater than 30 to 40 cohorts that have signed up to be active participants in these projects. The next slide, please. So I just wanted to just briefly tell you about 2 of the COVID projects. This one is led by Sarah Bauermeister in the Department of Psychiatry at the University of Oxford. Next slide, please. And it's focused on the mental health questions that have arisen from the pandemic, on an underpinning of stressors in our environments of unemployment, the economic downturn, social isolation, as well as the notion that individuals who had mental health disorders might be cut off from getting the kinds of care that they need, particularly with clinic closures and also data that suggests that there's a serious decline happening in individuals with underlying dementia, and it's clearly not unique to any one country. This is an issue of global import. Next slide, please. So the aims of this project are to begin to catalog the availability of cohort data on mental health assessments during and after the outbreak, mostly focused on harmonization in this aim of domain variables to build that will enable cross-cohort analyses in conjunction with the IHCC data standards, and it's taking advantage of the Dementias Platform UK's data curation initiative, which is composed of ontologies and standardization, characterization, and harmonization, and that's the DPUK CSIRV. Next slide, please. The other aims here are asking the questions around cross-cohort analysis to understand how the pandemic is affecting mental health symptoms or disorders over time, and which individuals will have risk or deterioration, which will have resistance, with a number of covariates that you can see listed here. And the second part of that aim is also to understand how social determinants of health might impact the impact of COVID-19 on mental health outcomes. Next slide, please. And the last aim is really to characterize neuropsychological and cognitive manifestations or complications of the infection, really looking at cognitive function and comparing pandemic times to reference periods in which there was no pandemic. So on the next slide, it just illustrates, I think, the 19 or so cohorts that are participating in this initiative, many from the US and UK, but also from other parts of Europe and South America. So clearly a global project. And the next slide is just wanted to highlight briefly a project who's led by Michelle Ramsey, professor of human genetics at University of Witwatersrand, and she's also president of the African Society of Human Genetics. Next slide, please. And keep toggling through this, the animation, keep going. Yeah, so this is really asking a question around the reasons why potentially prevalence of the pandemic or the infections are occurring at lower rates in African countries. And there's a number of potential hypotheses here that you can see in terms of immune protection, the population demographics, or even genetic resilience or susceptibility. The next slide, please. And these are some of the aims to really understand host genomic contributions to outcomes in South African patients, to really take full advantage of the partnerships locally in the continent to begin to understand the vulnerabilities to COVID-19, particularly looking at comorbidities, and to combine data from multiple African cohorts to enhance power. Next slide, please. This is one of the participating cohorts. I've mentioned it in the past, South African Population Research Infrastructure Network, which had already had put in place a number of platforms that would allow for bringing this type of research into the rural communities of South Africa. Next slide, please. And this just shows you one of those that was developed not for COVID-19, but for influenza and respiratory syncytial virus, really as part of a household study going into the communities for doing regional surveys and sampling on both cases, index cases, family members, and population controls. Next slide, please. So again, here, 5 cohorts on the South African continent are participating. This work is just getting started, and we're certainly excitedly looking forward to its success. Next slide. Then the one thing I wanted to leave you with is that we also have had work outside, proceeding in parallel to the studies I just mentioned to you that we recently funded. This is work being done with the Davos Alzheimer's Collaborative, which has been brought forward by the World Economic Forum and the Global CEO Initiative to really begin to understand the diversity and heterogeneity of Alzheimer's disease. Hakan Hakanarsson and Elias Serhouni are leading the scientific effort in this program for the IHCC and the DAC. The goal is to really collect and create a virtual cohort of 5 million people. As you can see on the left, many cohorts have already signed up to be participants. We're still defining how many individuals with Alzheimer's disease and dementia are harbored in them. But the goal is to understand the genetic heterogeneity, as well as to begin to understand the opportunities for liquid biopsies, as well as imaging and digital technologies to help us redefine some of the attributes of Alzheimer's disease and really enable the development of potentially novel diagnostics and therapeutics. Next slide, please. And then finally, just last week, we had our our semi-annual meeting. It was a virtual workshop in which we really tried to begin to undertake our future directions planning. It's very clear that we want to continue to have scalable demonstration projects, that we want to expand the atlas for cohort discoverability and access to the broader scientific community that would encourage and enable collaborations. We clearly see the opportunity to work with cohorts from low resource settings or low and middle income countries. Our goals also are to disseminate our work, to communicate well and to publish, and to entertain and carry out the possibility of cohort and enhancements, and finally to deliver a 5-year strategic plan, which it sounds like this the NCI Cohorts Consortium has successfully done recently. And then I think this is my last slide. Really just a second to last slide, just to remind you that we now have 110 global cohorts with 50 million participants approximately. And keep going, please. This is the website, which I encourage you to visit. And then on this next slide is the IHCC sponsors I mentioned before, the Wellcome Trust, the National Institutes of Health, and recently the Chan Zuckerberg Initiative. And I thank you for allowing me to open up this wonderful meeting of yours. I'm open to questions, and certainly if you have interest in the IHCC, you can contact me at jeffrey.ginsburg@duke.edu. Thanks very much.
Well, thank you. That was really wonderful and an overview for all of us. We have gone a little bit over our planned time, but we have ways to kind of make up some of the time. So I'm going to start you with two questions. The first one is a little bit easier. The question is from our audience. What are the funding sources for IHCC infrastructure and projects? I'm guessing that the question is that they would like to apply and propose projects. And so what would be the funding for that?
Right. So maybe I'll answer the question in both ways. As I mentioned, our sponsors, NIH and the Wellcome Trust in particular, have contributed equally to funding the 6 projects I mentioned in the last part of my talk. The Alzheimer's project is going to be funded through this Davos initiative. So we're looking for some projects to secure funding and bring it to the IHC, and other projects to come to the IC and request funding. So if you have ideas of things that, which I'm sure you do, and bring them to our attention, we're in the process, as I mentioned, of baking into a 5-year plan, which is really going to be a 5-year funding plan. We're going to go to the heroes in particular, and create a sort of a collective of funders to support the research that the IHCC is going to undertake. So this is the right time to begin to engage, have your ideas, and if we can, we'll try to bake as many of them that seem appropriate for our vision and mission into the plans for funding over the next 5 years.
Excellent, thank you. And they can get all that information about how to contribute and contact you through your email or through the website.
The website, but through the website too, yeah.
Okay, so my other question is, of these over 100 cohorts worldwide, how many of them are actually in a position to feasibly go back and get all those data about the experience of their participants with the pandemic? I mean, Dr. Collins mentioned about the response of people and how well the vaccine's going to work on everybody. So how many of all of these do you think will actually be able to go back into the field and gather those data?
Well, it's an excellent question about the feasibility. And because we have a lot of heterogeneity, and we're also working in places that lack resources and infrastructure in South America and Africa and some other parts of the world, we won't know until we get there. But the good news is that a lot of the cohorts that raise their hands seem to believe they have the wherewithal and to participate in these projects, seem to believe they have the wherewithal to participate, and we'll learn. I think it's one thing I'd like to mention. It's not really directly responding to your question, is the unique opportunity I think cohort data has to address something like a pandemic. When people are showing up in emergency rooms and data is being collected, it's acute data, and you have really very little knowledge of what preceded what data existed in that person's life before they were exposed and developed the infection. The cohorts have vast amounts of longitudinal data. You can really learn something from the past that actually will help you understand what's happening in the present and help you predict what will happen in the future. So it's something that I urge this consortium to think about, if you haven't already. I'm sure you have, is that these cohort data are very different than the kinds of data that are currently being collected by healthcare institutions on the acutely ill and their future manifestations of the disease. We have an enormous opportunity to leverage the pre-existing data in ways that they can't. But I want to, coming back to your specific question, I really don't know the answer yet until we sort of peel back the onion on the data. In each of the cohorts, we're not going to know. Last thing I'll say is that Wellcome Trust in particular has been proactive in this area. So really developing ways to work with cohorts of very diverse nature to help them ascertain the data that's necessary to answer the questions.
Hi, Dr. Ginsberg, this is Katie again. Thank you for a great presentation, and it's a great resource and a lot of good information there. So one thing I did notice is that there is still, for good reasons, stronger representation of European and North American and Asian countries. You have a little Australia in there as well. I was wondering, well, you did mention there was possibility of loosening some restrictions to get in some African and South American cohorts maybe, but can you talk about what that looks like and kind of if there's hope for getting in some more data from Black and African American or Black or African descent people or Hispanic and Latino descent people? And then what about Indigenous people as well? Is there a way to bring them into the fold?
So yes, absolutely. Let me answer your question in a couple of ways. One is that I want to make it clear that one of the tenets in our charter is that our goals are to enable cohorts, particularly in under-resourced settings, to be equal participants, both in terms of authorship and science, in any ways that we can. We're very sensitive to many of the lower resource setting cohorts feeling like they may have been, the right word is taken advantage of, but not really on. So really, in our funding plans, we aim to channel resources to the lower resource cohorts to enable them to be actively and equally participant in the research programs. As I said, we aim to relax criteria where the cohort has some pretty unique and highly desirable attributes, such as from South America or Africa. And we're also encouraging them to do some self-assembly, which I think would be very good for them as cohorts, as well as for their participation with groups like yours or group like ours. So for example, we know that there are several South American cohorts that are 10,000, 15,000, but nothing near 20,000. But in the aggregate, they may approach 50 or 60,000. And we're encouraging them to become a small consortium that can plug in, if you will, and participate with IATC. Similarly, in Africa, H3 Africa is I think trying to understand its overall organization going forward. And then there's the African Population Cohorts Consortium that's just getting off the ground. And again, we're working very closely with them in a lot of communication and trying to find ways to engage. Lastly, in our charter, we have members that meet the initial criteria, and then we have affiliate members, which pretty much is anybody else. And we're trying to make that distinction in part just to recognize the importance of the scale that we're trying to achieve, and but not to we want to be as inclusive as possible. So I would encourage any cohorts that are not in the stated criteria to contact us, and let's have a discussion about the ways to engage.
Okay, and I just have one more question before we end this session, Dr. Ginsberg. But because of the variation in government and regional responses to the pandemic, how are some of these studies going to be adjusting for the variation in sort of recommendations about the pandemic?
Yeah, that's a wonderful question. I guess I would want to ask you what are the variables that we should consider in terms of like understanding the variation of response. But I think to be honest with you, I don't know that in our haste to kind of get things started that we really gave that as much consideration as it probably deserves. But we know we are going to capture in some of the COVID-19 projects local regulations around social distancing, around curfews, around closures as much as possible. But we also know that that's a dynamic that's changing, particularly as we have this this surge that's going on. Things are changing rapidly, and we'll have to just make sure that we capture that data one way or the other and consider it as some potentially important covariate with whatever outcomes we're looking at. So I don't know yet the answer, but we're all learning about a lot of this in real time, aren't we?
Absolutely. And I think that was an excellent answer. We believe in science, and we understand that science is a growing, living thing. So with that, I'd like to thank you for your presentation. We are so grateful for your participating with us.